Emily. That was really nice. Yeah, dear all, can you hear me well? Can you see my
Yes.
slides?
Yes.
Okay, thanks. Many thanks for inviting me to talk about this research update on fertility in primary ciliary dyskinesia. Yeah, I'm Johanna Raidt, and I think I've been introduced really kind. Many thanks. As we already heard today, PCD belongs to a group of diseases which are called multiciliopathies. It is a multi-system disease, including different organ manifestations, and hallmark symptom is the recurrent respiratory tract infections. But this group of diseases is also associated with fertility problems. The sperm flagella of the male sperm cells is a specialized multicilium, which you can see here, which enables the sperm to propel through the female reproductive tract, for example. In addition, multicilia lining the male reproductive tract are also found in the ductuli efferentes to transport young sperm cells through the male reproductive tract. But I will come back to these mechanisms a bit later. This picture nicely demonstrates that sperm flagella and multicilia share a similar axonemal structure. The axoneme of multicilia and sperm flagella contain several different proteins and protein complexes. All of these proteins can be affected, and it is very important to understand that most genetic defects result in both sperm flagella and multicilia defects, but some defects are only in multicilia or sperm flagella. And here you can see how nice healthy sperm cells swim in this video. You already saw this picture from Marie and colleagues. Here you can see nicely again that sperm flagella and multicilia have a similar but not an identical structure. The sperm flagella, for example, contain different motor proteins which are sperm specific, and they are, for example, called DNH8 or DNH17. It is obvious that data on special tasks like fertility are very limited, especially in rare diseases. So we are very happy that Newman and colleagues this year published a review just recently summarizing the knowledge about the impact of PCD on female and male fertility. And it demonstrates very well that the fertility rate for men, for example, with PCD is unclear. They found 82 studies reporting male fertility outcomes. There were 192 men with PCD included, 167 men reported subfertility, and 25 men fathered natural pregnancies. There was only one cohort study included. This is the study by Van Aken and colleagues from 2017. They included 49 men with PCD, and 25% were fertile and 75% were infertile. There was no data on motility or ultrastructural assessment of the sperms, but the genotype was reported. This is in line with other reports, mainly reporting on single gene defects, that not all of the 15 known PCD genes affect sperm motility and/or function. Here you can see the included men with PCD in the Van Aken study. And of note, of these 37 men who were reported to be infertile, 15 benefited from assisted reproduction techniques and fathered 22 children altogether. As I already mentioned, there are more than 50 genes known to so far to be associated with multiciliopathies. Some of these genetic defects are clearly associated with dysmortile sperm. These genes encode, for example, factors that are responsible for the assembly of the dyneins. The dyneins are the motors of multicilia, but also of sperm flagella. And in these videos, you can see how affected airway cilia, but also how affected sperm cells look like in these mutant cells. Here you can see that the motile cilia are completely immotile, and also the sperm cells of these patients are completely immotile. Other gene defects are known to cause dysmorph and therefore dysmotile sperm flagella. Some of these gene defects were only known to cause male infertility because of these with this dysmorph sperm cells. And some of these genes were also only known to cause PCD. Now we know that there are gene defects which cause both dysmorph and dysmotile sperms and also PCD. This is, for example, true for the hydine mutants or the SPEF2 mutants. And in this videos, you can see how hydine sperm cells only move a little bit and they're really dysmorph. And again here, compared to control sperm cells. For male fertility, not only the sperm cells, but also the motile cilia of the male reproductive tract are of great importance. It could be shown that DNH5 mutations lead to immotile cilia of the ductuli efferentes. The ductuli efferentes connect the epididymis with the testes, and they are responsible for the transport of young immature sperm cells to the male reproductive tract. Dysmotility of the ductuli efferentes cilia result in sperm retention and therefore can lead to subfertility in PCD patients. Here you can see ductuli efferentes cilia of a mice, which are healthy and therefore here are moving nicely. And with this slide, I'd already like to summarize and give some additional aspects on fertility in men with PCD. We've learned that variants in dynein pre-assembly genes, as you can see some examples here, lead to complete immotile sperm and therefore are associated with a high level of infertility. We've also learned that variants in genes resulting in central pair defects, for example, SPEF2 or hydine, or tubular disorganization combined with inner dynein arm defects lead to dysmorph and dysmotile sperm and are associated with a reduced and very variable fertility. We've also learned that DNH5 variants result in reduced sperm count due to sperm retention and therefore cause variable fertility. What I not talked about is that all published data on men with genetic variants in SPH4A, for example, show that they are fertile and that variants in genes encoding components involved in ciliogenesis result in absence of sperm in the ejaculate in the mouse model. And with this, I'd like to conclude on this. It is not sufficiently understood which PCD genes and genetic variants affect sperm flagella and motile cilia of the efferent ducts and therefore impact male fertility in PCD. There are increasing number of studies, which is great, investigating the influence of individual genes and genetic variants on sperm cells. And we've learned that men with PCD are not necessarily infertile, and that there might be a potential combined effect of genetic PCD variants on sperm development and transport. And it is no doubt that systematic cohort studies are needed, for example, by putting fertility data in national and international registries to perform genotype-phenotype studies, and that there is a high need of translation of these study results into the clinics to inform affected individuals and the medical staff and guide the clinical care of affected individuals. And if ever possible, to establish a multidisciplinary team, including also fertility experts, for example, andrologists or gynecologists. And with this, I'd like to come to the female part because the female reproductive tract is lined with multiple motile cilia, for example, in the oviducts, which are here. I come to that later. Unfortunately, there is very less data on female fertility and underlying mechanisms. One reason, of course, is that the female reproductive tract is not so easily accessible and analyzable compared to, for example, sperm cells. By reviewing the literature, you can get the impression that female patients with PCD are, of course, infertile or at least subfertile, but it is not known if a genetic defect leading to dysfunction of airway cilia does also affect the cilia of the oviduct. And here again, the study of Newman and colleagues is very helpful to review the current literature. And in summary, the fertility status of women with PCD is also unclear. In this study, they found 37 publications reporting on fertility, female fertility outcomes in PCD. There were 119 women with PCD included. There were 71 women reported subfertility and 48 women reported natural pregnancies. As for males, there are only limited systematic studies. For example, the study also of Van Aken et al. With 36 women with PCD and 61 of them reported to be infertile. And there was a study from our lab some years ago with 9 women with PCD and severe respiratory phenotype who were 100% fertile. And again, as for males, for the 50 or more than 50 PCD genes known so far, there are only limited data for fertility in women. Here you can see again the Van Aken study where 36 women with PCD were included, 14 reported natural pregnancy, and no ectopic pregnancies were reported in this study. There were 8% miscarriages, and 22 females were defined as infertile and 6 got pregnant after assisted reproduction. Here is the study from our lab. We reported some years ago on 9 women with PCD and genetic variants in common PCD genes, which lead to severe dysfunction of airway cilia, resulting in classical respiratory disease in these women. And all of these women gave birth to 1 or more children without reporting fertility problems. This is an overview, somebody is painting on my slides. This is an overview of the female reproductive tract. We have the 2 oviducts connecting the 2 ovaries, which you can find here with the uterus. In the video, you can see a longitudinally sliced human oviduct, and you can see, I hope you can see nicely, the movement of the motile cilia along the surface. What we also looked at in our study is the particle transport experiment, and we could show that the motile cilia in the oviduct create a directed fluid flow from the lateral edge of the oviduct towards the uterus. I hope you can see the small red arrows which are guiding the way. And with this slide, I'd like to summarize also the female part and give some additional aspects on fertility in women with PCD. There seemed to be a coordinated beating pattern, the same as an airway cilia, and there seemed to be a directed transport towards the uterus. There seemed to be a similar composition of motor proteins, which I haven't shown here because of time reasons. There are reports about female infertility in PCD, but also on women with PCD and severe dysmotile airway cilia experience natural pregnancies. There are ectopic pregnancies reported in women with PCD, but do not seem to occur frequently. Miscarriages are rarely reported, for example, in the only cohort study, but there was no increased rate reported. And in conclusion, that means that the potential motility defects of oviduct cilia do not appear to exclude success reproduction, but there are no analyses of affected oviductal cilia reported. And we've learned that women with PCD are not necessarily infertile. There is a high need for well-designed prevalence studies for systematic assessment of female fertility in PCD, for example, by entering fertility data in national and international registries and databases for genotype-phenotype correlations. And it is necessary to translate all the study results into the clinics to inform the affected individuals and also the medical staff and guide clinical care. And whenever possible, to establish a multidisciplinary team, including fertility experts, gynecologists, and urologists, etc. Yeah, and for time reasons, I could not go into detail to the management of reproduction and pregnancy, but there are some nice studies. For example, here, the study in 2020, it's a general study on women with airway diseases and the management of reproduction and pregnancy. And there are some PCD-specific studies out now. Just recently, this study in the middle, really nice summary about fertility care among people with PCD, and also the study on male infertility from 2 years ago. And with this, I'd like to thank all the great team of Münster and the Department of General Pediatrics and the laboratory of Professor Omran. And I'd like to thank you all for your attention, and I'm happy to take some questions, or I'm happy for everybody to get in touch maybe later.